Thank you. Thank you for that very kind introduction, and it really is a privilege here to speak with you today. The conference has been great so far. It's always nice to hear what other countries do outside of what we do. I will say that sickle cell disease is a growing problem globally. In the United States, we have about 100,000 patients with sickle cell disease. At my institution alone, we care for 1,000 active patients just in our hospital alone. So today, what I want to discuss with you are new tools to personalize transfusion therapy for sickle cell disease. And what I hope that you'll take away from today is that there's still a fair amount of work if we want to provide them with precise blood matching, and I think that's in the near future. So I have nothing to disclose. Today we'll discuss alloimmunization risk in patients with sickle cell disease, the role of Rh genotype matched transfusions, and I have a few slides at the end about the use of induced pluripotent stem cell derived red blood cells and how this could be a renewable source of red cells. So most patients with sickle cell disease will have received a blood transfusion by the time they reach adulthood. And as you just heard, prevention of a first or recurrent stroke is the most common indication for chronic transfusion therapy. More recently, it's become a prerequisite for some curative therapy trials, whether it's for bone marrow transplant or for gene therapy. They are oftentimes required to have a 3 to 6 month period of red cell exchange prior to curative therapy. However, alloimmunization remains a frequent complication, and acute and delayed hemolytic transfusion reactions are among the most challenging complications of transfusion support in this patient population. The primary reason for alloimmunization is likely recipient donor red cell antigen mismatch. So the patient receives donor red cells that have antigens that the patient themselves lack. However, in the past decade or so, we've had increasing awareness that Rh genetic diversity, both in our patients and in Black donors, does contribute to the Rh alloimmunization risk for these patients. And then lastly, compared to other chronically transfused populations, chronic inflammation and immune system dysregulation also plays a role. So the chronic hemolysis leads to free heme and hemoglobin, and this increases your inflammatory pathways. And patients who are transfused episodically, for instance, when admitted for acute chest syndrome or for stroke, are at higher risk of developing an antibody in those settings than when they're transfused on the outpatient setting. The American Society of Hematology, now about 4 or 5 years ago, set out to produce some clinical guidelines, and one of the 5 areas of interest was for transfusion. And here, I'm just showing one recommendation where ASH does put out that for patients with sickle cell disease, it's a strong recommendation to provide prophylactic red cell antigen matching for Rh, big C, big E, or big C, big E, little e, and K antigens for all patients with sickle cell disease. And it might surprise you, but in the United States, not all university systems and not all hospitals provide CEK match blood. It's really primarily at the larger institutions who care for many patients with sickle cell disease. And this was primarily made a strong recommendation because single-arm studies, when we pooled the data, showed that there's a significantly lower rate of alloimmunization with Rh and Cal or with extended matching. So for ABO dematched alone, patients make approximately 2 antibodies per 100 units transfused. If we provide Rh and K matched cells, it decreases to about 20% of that. So 1 in 250 units, an antibody will be made. Now, extended matching for the Duffy, Kidd, big S, little s, does reduce alloimmunization further. Just finding the number of units to provide this patient population for extended matching becomes even more difficult beyond CEK matching. So I just want to share a case that we saw years ago at Children's Hospital of Philadelphia. This is a 5-year-old male with sickle cell disease type SS and who presented to the emergency room with fatigue, jaundice, and hemoglobinuria, as you can see to the right. He had been transfused 1 unit of O positive Rh and K matched red cells 10 days prior, and his pre-transfusion hemoglobin was 6.5 grams per deciliter, and his antibody screen was negative at that time. When he presented to the emergency room, his hemoglobin was 5.6, so lower than his pre-transfusion hemoglobin, and subsequently in the next 24 hours natored at 4.2 grams per deciliter. His hemoglobin A on quantification was 7%. This is 10 days post-transfusion, and by the next day, it was 0. So of course we suspected a delayed hemolytic transfusion reaction. However, he was DAT negative, and his antibody evaluation showed weak reactivity to all cells except for one on a panel of at least a dozen panel red cells. It was a little bit delayed, but we did make the antibody identification. The patient had made an anti-D. However, the technologist who was doing his antibody evaluation wasn't paying attention to the D because he's D positive and wasn't looking for an anti-D. So initially, when everything had weak reactivity, they thought we were likely dealing with a pan-agglutinin or a warm autoantibody, which we oftentimes do see in patients with sickle cell disease. Nonetheless, we performed Rh genotyping, and this was years ago before we were doing it for all of our patients, and his genotyping showed that he was homozygous for the RhD variant DAO4, which results in partial D expression on his red cells. So he has D antigen on his red cells, but lacks a common D epitope, and thus his anti-D is an alloantibody and not an autoantibody. And I bring this case forward because it's a patient who did receive CEK matched red cells, but we're still not precisely matching our patients, and we need to do more. So we looked back at our own institutional history of red cell alloimmunization because in the mid-1990s, Dr. Kim Smith-Whitley, who's a hematologist at CHOP, started with the American Red Cross and the Sickle Cell Disease Association of America a donor program that specifically recruited Black donors in the Philadelphia region. And when they donated, they would take that blood and put it aside at the Red Cross for children with sickle cell disease, either at Children's Hospital of Philadelphia or St. Christopher's Children's Hospital in Philadelphia. And we had been doing this for nearly 2 decades, and no one had looked yet at what our rates of alloimmunization were. So we went back and looked, and we had a really surprising finding that the majority of our antibodies were still directed against the Rh blood group system, which are the bars shown in red and boxed. Our rate of alloimmunization was 1 antibody per 250 units transfused, and that's important because we provide CEK matched red cells from minority donors, and the rate of alloimmunization is the same as other institutions who provide CEK matched donor units, but the types of antibodies we see are slightly different. Shown in the black bars are the other antigens to which patients may get alloimmunized, and consistent with what we expected by giving them donations from Black donors, we did see decreased numbers of anti-KID, anti-Duffy, anti-Big S. As I mentioned before, 2/3 of the antibodies were directed against the Rh blood group system, and as I mentioned previously, we now appreciate that it's the Rh diversity in our patients and in our donors that contributes to this rate. So this just shows the Rh variants that are frequent in patients with sickle cell disease and Black donors. There's over probably 500 different alleles that have now been reported in the Rh blood group system, but we do see is that in our patient population and in our Black donors, at least in the US, where much of the African immigration occurred is from West Africa, we see that it's the same 20 RhD alleles and the same 20 RhCE alleles in general. And the frequency of the alleles in both the patients and in the African American donors was really very similar. So to just back up and just explain a little bit more about the Rh blood group system, as you know, there's the RhD protein and the RhCE protein. And when we have variant alleles, we oftentimes see that they will encode partial antigens. So on the left is the example that I spoke of before, the DAO4 allele. And in this allele, there's 2 amino acid changes that result in a partial D antigen being expressed. A very common allele we oftentimes see in Africans is this RhCE CE733G. And this too results in partial little e and partial little c antigen expression, but it also causes lack of this high prevalence Rh antigen, HRB, and these patients express another Rh antigen that's considered a low prevalence antigen in the general population, but actually 30% of Blacks have VS on their red cells. So this complicates how we think about alloimmunization in patients with sickle cell disease who are getting donor blood from the African population. And while our standard typing reagents do recognize the conventional standard Rh antigens, D, big C, big E, little c, and little e, they don't distinguish when a patient has a partial antigen, and they don't distinguish the variants. So Rh variants are prevalent in patients with sickle cell disease and Black donors who are needed to support CEK matching programs. So when we looked back at the Rh alloimmunization, we saw that patients aren't truly Rh matched when you do serologic antigen matching. So overall, 30% of the Rh antibodies that we saw could be attributed to the patient's Rh genotype, like the case I showed you. The patient's D positive expresses a partial D antigen and makes anti-D, which is an allo antibody. However, the remainder appeared unexplained. So our hypothesis is that the Rh variants on donor red cells, since the majority of donors that they saw were from African American donors, is an underappreciated risk factor. So again, both the donors and the patient's Rh diversity contributes to this Rh alloimmunization. And you can see here on the chart that, for instance, anti-D really only occurred in patients that are D positive. We have one patient who's D negative and probably saw a DEL unit. And if you look at the anti-little e's, since most patients are little e positive, those patients all received likely antigen positive units because they were getting units from Black donors. However, I just want to bring up the point that recruiting Black donors remains really important. So many people, when we first came out with this data, said, are you still providing your patients with African American donor units? And we said yes, because we really can't support a CEK matching program without that very valuable donor population. As I mentioned, this was a program we started in the mid-1990s, and I think as the US sees more and more admixture, that's more and more reason why we need to go to genotyping to match rather than relying on a donor to self-identify as a certain race. Recruiting and retaining Black donors is fundamental to an adequate blood supply for patients with sickle cell disease in the US and obviously abroad as well. And similar to what was brought up this morning, the challenges that we have are that African American donors typically have a higher donor deferral rate, either due to anemia or hypertension. And there are other challenges, such as fewer drives in Black communities, so getting drives that are in the community will likely help, as well as lack of awareness of the importance of their blood donations for patients with sickle cell disease. So the current challenges right now is that alloimmunization remains problematic despite phenotype matching strategies. Antibodies against the Rh blood group system are the most common and often the most complex to identify. And to improve red cell matching and safety, we really need assays to, one, identify those at highest risk, two, improve accessibility of blood group genotyping, especially Rh, and three, we need access to rare red blood cells with specific Rh phenotypes to aid in antibody identification, and ultimately, these could be used for transfusion as well. So how do we use genotyping currently? So we've moved away from serologic antigen phenotyping at my institution. We now get a genotype predicted extended red cell antigen profile performed at the first visit. The one that we use is called the human erythrocyte antigen. It's a B chip that is actually, for the US, an FDA approved assay for red cell profiling. And it provides antigen data on 35 antigens. So typically when we serologically phenotype, we phenotype for about 12 to 15 antigens. So clearly we're getting more with the genotype. It's one used to guide prophylactic Rh and K matched transfusions. But the other way we've found that it's been really helpful is that it informs antibody evaluation when a patient has a new positive antibody test, because now we have information on 35 different antigens rather than 12 to 15. In addition, we get a comprehensive Rh allele identification for all patients with sickle cell disease. So this is performed at their first visit along with the HEA genotype. And from a cost perspective, our blood bank did an analysis, and for the price that we paid for the HEA genotype as well as a comprehensive Rh genotyping, it really was a wash in terms of how much we pay for our red cell reagents and keeping techs up to date on the serologic phenotyping. And again, this informs antibody evaluation when a patient has a new positive antibody test, particularly in the Rh blood group system. We've also used it to guide how we prophylactically match patients. So for certain patients who are considered to have high-risk Rh variants, we provide them with prophylactic antigen-negative red cells. So for instance, patients with alleles encoding partial C, which here I show a very common allele that patients have. So actually on their RHD locus, they have 4 exons from RHCE that have translocated into the D gene. And this is a D-negative allele, but expresses partial big C. And patients with this partial big C type strongly big C positive with typical monoclonal antibodies that you use for antigen typing. However, if the patient does not have an RHCE allele that expresses a conventional big C antigen, we provide them with big C negative blood. And we've been doing this for 7 or 8 years now, and we have not seen another patient make an anti-big C when they have this partial big C allele. Well, it's really a partial D allele expressing a partial big C. We also, more recently, are also providing patients with variant RHD alleles that encode partial D antigen with D-negative red cells. And that's always been a little bit difficult because we know that if we provide them with D-negative blood, that oftentimes they're going outside of the Black donor inventory, simply because less than 5% of Black donors will be D-negative. And so that may put them at increased risk for making antibodies outside the Rh system, like anti-JKA or JKB. For patients who were unable to get the comprehensive Rh genotype, because in the US it depends on your insurance, we recommend that the Rh genotype still is obtained for those who are Rh immunized or who are multiply immunized. But if they haven't received transfusions and they're not going on chronic transfusions, if their insurance doesn't cover it, we won't obtain it, just because the cost would then be borne by the family. But I would say in general, over 95% of the insurances do cover the genotyping. It's also important to note that Rh genotyping is not standardized amongst reference immunogenetics laboratories. So the way we have approached it with the New York Blood Center, who has been our collaborator with all the Rh work that we've done, is that the genotyping is done using DNA arrays that target common variants. So the RHD and RHCE chips have 35 RHD and 25 RHCE single nucleotide changes or insertions that are identified. However, it doesn't cover some of the most common SNPs, so these need to be supplemented with PCR-based laboratory-developed tests. So they test for other frequent changes not on the array, importantly the RHD exon 8 change and an RHC exon 2 change that exon 8 change has an allele frequency of over 10%. So it's really important that when you get Rh genotype, you have to realize that different labs do it differently. In addition, RHD zygosity tests are performed as well to know if patients have hemizygosity or homozygosity for D alleles. So for Rh molecular red cell matching for sickle cell disease, we do think that Rh alloimmunization could be avoided by prophylactic Rh genotype matched red cells for transfusion. And it's likely feasible with a robust Black donor pool. So as I had shown you previously, the frequency of the alleles is very similar amongst our patients and donors. And we have genotyped over 1,000 patients with sickle cell disease, and we compared it with 600 donors as they came in. So not culled from their donor registry, but literally 600 consecutive Black donors that had presented for donation. And we had performed electronic Rh genotype and K matching for a population of 200 patients that had been transfused at our institution over 4 years, requiring 28,000 units over that 4-year period. So you can see the types of resources these patients require. And we compared that using the observed Rh genotype frequencies. And what we found bioinformatically was that unit demand could be met with twice the number of Black donors screened per day compared to serologic matching. So what we screen per day using serologic CEK matching to provide those units to those patients, if we screen twice as many, we would be able to find them genotype matched donors, which is not that many more. And if you think about doing it on a national basis, you could make it much more efficient. The cost of genotyping is still a major barrier, but I think that's something that will be overcome in the next several years. So current methods for Rh genotyping include commercially available Rh SNP arrays. As I mentioned before, our patients have been genotyped using the BlooDHIT, which covers 34 RhD and 25 RhCe SNPs. There's also the BlooDHIT by Grifols, which covers a few RhD and RhCe SNPs, and oftentimes these are supplemented by PCR-based assays. There's also automated red blood cell genotyping algorithms that have been applied from whole exome sequencing and whole genome sequencing data, and this is just important because it provides proof of principle for NGS-based assays. There's BlooDTyper that was performed by Bill Lane and Connie Westhoff, which looks at red blood cell and platelet antigens, but doesn't cover so much the variant antigens as the conventional antigens. And then RhTyper is something we worked with Dr. Zhang at St. Jude's Research Children's Hospital, which specifically takes whole exome or whole sequence data and analyzes it to provide the Rh alleles that the patient has. And so this is an example from the paper. We used RhTyper in 884 patients with sickle cell disease who had already gotten whole exome or whole genome sequencing through other studies. And overall, we detected 41 RhD and 28 RhCe SNPs or indels that corresponded to 38 RhD and 28 RhCe alleles. And so this is just showing the different SNPs that we found in RhD and the different SNPs that we saw at RhCe. And so the bigger circle will show, for instance, 657 of these 884 patients had this SNP. And importantly, again, as I mentioned, we see the same RhD and the same RhCe alleles over and over again, despite having hundreds reported in the literature. And in this study with 884 patients, we found one novel RhD DAO allele, suggesting that most Rh alleles in this patient population are known. So NGS assays still are needed to provide wider application of Rh genotyping of patients and donors. The benefits of NGS is that the number of markers is not limited. It should detect all polymorphisms, including null alleles, novel mutations, gene rearrangements, and copy number analysis can be performed. The limitations are that read alignment is difficult for duplicated and homologous genes. So the RhD and RhCe system alleles are a duplicated gene family, highly homologous. So when you have short reads, you don't know necessarily should you map it to RhD versus RhCe without having enough sequence beyond the 200 to 250 base pair reads that you get. Phasing of SNPs that are thousands of base pairs apart also is not possible for haplotype assembly and allele assignment. However, I think in the future, we've already seen proof of principle using whole exome sequencing, whole genome sequencing, and long read sequencing platforms that we can pretty accurately determine the Rh alleles of patients with sickle cell disease using these NGS-based assays. And in the future, probably targeted Rh NGS assays will be available using very customized analysis pipelines that specifically are designed for the Rh allele variants that we see in people of African descent. And so using that along with copy number analysis, I think we can overcome the challenges that we have for NGS-based assays for Rh and will make it a lot more accessible for patients and then as well for blood centers to be able to cost effectively genotype their donors. So with that in mind, we embarked on a pilot feasibility study to provide Rh genotype matched red cells to patients on chronic transfusions. This is Stacy, our research coordinator, and Connie Westhoff and Sunitha Vijay at the New York Blood Center, with whom this work is being done collaboratively with. And what they are doing is they're screening Black and Hispanic donors with PCR-based assays that look for specific alleles, and then after the screening, performing comprehensive Rh genotyping, the same as what was done for our patients, for the relevant donors before they ship us the units. And our approach for Rh genotype matching is that For instance, if this is the patient's genotype, the donor match could either be an exact Rh match where all 4 alleles are identical to the patient's, or the donor could be an Rh haplotype match where they're homozygous for one of the patient's haplotypes, or it could be a Rh allele match. So any 3 of these would be a sufficient donor match. We also said that it was okay to do a less restrictive match in the sense that DAO0 and CE48C are very high frequency in Africans and have never been shown to lack an epitope, even though it's very indirect evidence, but we hypothesized that DAO0 and CE48C would be equivalent to the conventional alleles, so a less restrictive match would allow us to substitute RHD for DAO0 and conventional little c, little e for the 48C variant. And by doing this, we so far have had 10 patients who are enrolled in our study. These are the genotypes shown here on the bottom, and you can see that they're quite varied. And so far, we've been able to provide the majority as an exact Rh genotype matched, which you can see in green. And in the blue are patients where we did substitute the RHD and DAO0 and the CE with the CE48C. And thus far, we haven't had the feasibility study was seeing if we could find the donor matches and making sure we could find the donor matches in time for their transfusion. So these are all patients who are chronically transfused. So to be fair, we know they're coming again, and we're looking for their donor units. And we're applying the same rules of having to have cells that are less than 21 days of age, hemoglobin S negative, in addition to their CEK match. And if they have an alloantibody to an antigen, we of course still provide them antigen negative units. And thus far we haven't had any delays in transfusion, and it's only been about a little bit over 200 units so far, but we haven't had any patient who's made an Rh antibody. But as I mentioned, we only see 1 in every 250 units, so we'll have to transfuse many more units before we can definitively say that we're avoiding Rh alloimmunization. So an Rh screening tool can be designed to identify the relevant donor genotypes and eliminate other donors. And importantly, because a genotype is a genotype, once they genotype the donor, it's in their system, and we have had cases where we're using the same donor again who's been genotyped previously. We did embark on this just as the pandemic started, but despite the challenges related to blood shortages, Rh matched units were identified for all enrolled subjects with no transfusion delays. Efficiency will definitely be enhanced with scale-up of patients requiring Rh genotype donors and improved NGS-based assays. And then, again, future studies are necessary to test the effect on Rh alloimmunization. The other thing that's really difficult for patients with sickle cell disease sometimes is the identification of compatible blood. And because finding compatible blood for patients with sickle cell disease can be particularly challenging because we don't always have the ability to identify the exact specificity of the antibody that they seem to have a positive antibody screen for. Antibody specificity also oftentimes may not be quickly identified, so oftentimes we'll have a patient with a new antibody on antibody screen, but then it takes days before our reference laboratory can give us the antibody identification if they find the antibody specificity. And this is really due to a lack of appropriate reagent red cells, and also because many Rh antibodies are complex, particularly to high Rh prevalence antigens. And so rare donor red cells with specific Rh phenotypes are needed. And so we hypothesize that induced pluripotent stem cells may provide an alternative source of cells because it's a renewable, unlimited source of stem cells, and they're amenable to genetic engineering. So this was a project done primarily by a student in my lab, and what we did was we took iPSCs, either we reprogrammed uncommon donors that we received from the New York Blood Center into iPS cells, or we gene-edited iPS cells. So we took all the iPS cells we already had, and we did ABO and D genotyping and looked for O-negative phenotypes. And then what we did was we gene-edited to produce rare red cells. So on the right, you'll see we made an Rh null cell by taking a O-negative cell and then knocking out the RHCE allele. And we then use some gene targeting strategies to then put back in the RHD locus and make D minus cells. And so many of you know that there's only a few people walking on Earth with the Rh null and Z minus phenotypes, but for antibody identification, these can be really useful. We also use genetic engineering to put in the cDNA so that we would produce red cells with these two D variants, GOA and DAC positive. And then we made HRS negative, HRB negative, and VVS positive red cells by reprogramming donors who were genotyped that would have that red cell antigen phenotype. And then we produced the red cells, and then we use these red cells on gel card assays. So the way we differentiate the iPS cells is that we do an embryoid body formation, which basically leaves us at about day 7 to day 8 of culture with these progenitors that actually are shed from the embryoid bodies. And then we put them into an erythroid-specific mix to make those hematopoietic progenitors into red blood cells. And so this is a cytospin of the red cells. They are bigger. This is also smushed up against the slide. If you look at it and you measure their actual size, they're about 20% bigger than a donor red cell from donor units. So the enucleated cells on this cytospin are donor-derived red blood cells, and the nucleated ones are the iPS-derived red blood cells. And this is just an example of flow cytometry where we looked at the Rh expression. This is a donor-derived red cell. This is our untargeted OD positive but CE negative red cells, and this is the Rh null where you see that there is no Rh expression on the red cell surface. So then we use these to see if we could identify antibodies. So in this case is an example of how the IRBCs could be used to distinguish antibodies against high prevalence Rh antigens. So on the left are donor-derived red cells of the typical 3 Rh phenotypes that we usually do an antibody screen with. And you can see that this patient's plasma reacted with the R1 and the little r. Oops, let's go back for a second. Little r cells, but did not react here. And so these two cells are both little e positive and also HRS positive. And here, this would suggest that the patient possibly made an anti-little e. However, if we test it with our Rh null cells, you can see, and with the D minus, you can see that there's no reactivity, so it suggested it was against the CE system. But here we have an IRBC that's E positive, HRS negative, and HRB positive. And here we have an IRBC that's E positive, but HRS positive and HRB negative. And so this would suggest that this is really an anti-HRS. Conversely, we can use IRBCs to detect antibodies against what are considered low prevalence Rh antigens in the general population. This is an example where this patient plasma was reacted with the common Rh phenotypes, and it suggests that there's no antibody present. But you can see here that when we tested it against 3 of our uncommon phenotypes, GOA positive, DAC positive, and VVS, it reacted really nicely with the red cells expressing GOA. So iPSCs can provide an alternative source of red cells, but scale up and storage, the shelf life, remain challenges. Rh-engineered IRBCs provide a novel tool to aid in complex Rh antibody identification, and future studies aimed towards storage solutions and enhancing cell yields will be needed to commercialize this. So in summary, the future of transfusion therapy for sickle cell disease, Rh genotype matched red cells likely are feasible and could minimize Rh alloimmunization for this patient population. It would require a robust African American donor pool or African Irish donor pool. In vitro derived red cells with rare antigen phenotypes may facilitate antibody identification and exact specificities. And it'd be really nice if we could get these to be accessible to hospital blood banks to perform the testing in-house. So the combination of these new tools can allow much more precise red cell matching and could, beyond sickle cell disease, be a paradigm for all patients requiring red cell transfusion. So I just want to acknowledge people in my lab, those who helped with the IRBC project, and our many collaborators for both of those projects. And I'm happy to take questions.
You're going to stay there, Stella, yeah? Right. Listen, yeah, thank you so much, Professor Chou. You do not disappoint. Any questions for the speaker? And Grace, you're going to work the room, yeah? Thanks.
Stella, that was absolutely wonderful. Thank you so much. Just wondering, no pressure, but what is the time frame on the commercially available IRBCs? They look wonderful.
I think it'll still be years, honestly, because finding the right storage solution has been a problem. We've tried all the typical storage solutions that blood centers use for reagent red cells and for storage of red cells, and they don't last for more than a week, typically. And so once they start to senesce, then you just get non-specific agglutination. So I think finding the right storage solution will be key for us.
Years.
I think so, because they're so different from donor-derived red cells, but we're trying to work with Grifols to see if they can help us find a storage solution.
Yeah, just sort of waiting. We have an advantage here, Stella. The patients are all molecular genotyped, which is unusual. Like you mentioned your institution, your patients are typed, so are ours, but obviously we need to move into the molecular space. We'll talk about that maybe internally in IBTS. We don't have molecular genotyped blood donor infantry routine. I guess that's our problem. We do transfuse, whatever about the partial Ds and the DOWs could be as high as 20%. We don't see anti-D formation, which is interesting, you know, but even though we're transfusing Caucasian blood into these patients, but we haven't done a proper structured analysis or review.
Yeah, I think it's part of the anti-D that we see is definitely the donors. So we see actually, this is like the nitty-gritty, but the patients who actually have partial D and form anti-D, they usually form that anti-D with a relatively low number of D positive exposures. The patients who make anti-D but have conventional D or DAO0 themselves, sometimes they form that anti-D after, say, 200 exposures. And the anti-D typically doesn't persist as long as the patients with partial D. So they all evanesce, but the partial Ds, when they make anti-D, they might continue to demonstrate that anti-D for months to a year or two. The patients who make anti-D from being exposed to some Rh variant on donor cells, we see that they're quickly evanescent, that they only last in the plasma for just a couple of months. So they're definitely different.
Sorry, Dermot.
Thank you very much for a fantastic talk. I'm very curious about your patient cohort and the chronic transfusion program, that you're actually getting enough of the genotype matched blood, or even just the partially matched. So, for example, in patients who might be receiving a large volume red cell exchange, are you giving a mixture potentially of compatible and potentially partially compatible red cells?
So for our patients that are getting exchanged, actually there was one patient on that graph I showed who was getting exchanged and getting the Rh genotype match. So that patient was relatively small though and was getting 4 units per exchange transfusion, but we were able to find him Rh genotype matched cells. Typically for our exchange program, we provide serologic Rh and K matched cells, and typically we match for the big C and big E, and then match for anything they've made antibodies to. So most times they're getting, serologically matched cells, but I think that it is feasible to find them genotype matched cells if we could scale up the genotyping of donors.